I will continue in the same line of Professor Stephen Mayfield have done, and I will call him father of modern algal biotechnology because he is the single person in all over the world who has mostly contributed in algae and showed the path to like our next generation. So thank you to him also. So algae is the same like photosynthetic organisms, but it many people don't believe it's like a plant. So for regulatory agency, it's quite difficult whether to consider it's plant or some different organism. So I will be putting some highlight on different aspects here and showing some work related to our research. So if you see that the in globe like the US is the leading in the algal biotechnology, and if you see the India don't have even a mark on the worldwide in the algae business. So maybe in hope in future like Reliance and CPCL and many department of biotechnology will make the footprint in the algae biofuel or products. So why we want to do on algae? If you see the diesel in India is like 3 times over the petrol, and in petrol you can add the ethanol, but for a diesel you need some substrate like biodiesels kind of things. So algae could be one substitute for that one. And if you see that if you burn the fossil mineral diesel 100%, you get the 100% carbon release. But the algae biodiesel just produce 12% carbon. So the carbon footprinting is way less than comparative to the biodiesel produced from the crops or in compared to diesels. So, but the problem is with algae, the disadvantage that high production cost due to low lipid, low biomass, and harvesting process. So these are the issues we want to deal to reduce the cost. So in first example, we have genetically modified this Chlamydomonas, model species with the Brassica nepes D-get2 genes, which is responsible for neutral lipids. And so, and we have seen that Chlamydomonas hardly produce any lipids, are filled with this by expression of this D-get2 gene of Brassica nepes into the algae. They're producing lots of lipids. So a lipid like neutral lipid was produced from 3% to the around 7%. So double the amount of lipid can be produced using the GM technology. So that could be helpful for reducing the cost. Second, like carbon dioxide is a big problem all over the world. So if we can genetically modify the algae to sequester more carbon dioxide, then we can help the environment as well as convert this excess carbon dioxide into the like starch or lipid biosynthesis. So this is the Rubisco, and Rubisco is present inside in the chloroplast surrounded by the pyrenoids. So here what we have done, we have taken two membrane transporter LCIA and LCIB. They are located in chloroplast envelope and stroma. So we have introduced this LCI in this membrane transporter in the algal systems, and to sequester more carbon dioxide, because Rubisco is slow enzymes. If it doesn't have much carbon dioxide, then it start doing the photorespiration. So to enhance, to concentrate the carbon dioxide, we have re-networking this carbon fixation pathway, and this is the just some studies which has been confirmed by by a student that these are the transgenics lines, and further the more studies. So this is the interesting. If you see the wild type, the starch layer is very small in around the pyrenoids, but in transgenics the starch layer is very thick. And you see that within 5 days, the algae, transgenic algae producing the double the biomass compared to the wild type. So these genes are helping to capture more carbon dioxide. So that's the one way to go forward. Uh, and the lipid also, if you see that in wild type, uh, the lipid is not much, but in transgenics, lipid has been like 50% of the cell has been filled with in the transgenic cells. And you can see the biomass also has been increased. The dry biomass produced like from 21 in wild type, in transgenic it has been increased like 3 to 4 fold. Same way lipid has gone up like 2-fold up. So, Professor Mayfield has said that there's many challenges in the algae. So it's not simply like one step we do and we are done. We have to tackle many steps. We are working on the genetic transformation tools like CRISPR-Cas and increasing neutral lipids and developing CCM mechanism to capture more carbon dioxide and getting directly the FAME biodiesel from the wet biomass rather than drying, and using the endophytic fungus to enhance the lipid accumulation as well as biomass, and making it easy harvesting just like a sieving kind of process. And then we use wastewater of our institute to grow these biomass of algae, and we have developed these efficient photobioreactor which run on the solar panels. So back to the regulatory concerns, the green algae and cyanobacteria are the photosynthetic organism. So it may be difficult for the regulatory agency how to control them, whether they come into the different microbes or they are undercome the Plant Protection Act. And this is the one example of a US again in Hawaii in 2005 that a company wanted to grow these genetically modified chlamydomonas to produce some pharmaceutical, but they could not get the license because the regulatory agency could not understand what could be the process to regulate such kind of organism. So this is such kind of difficulty, and moreover there's a more hypothetical risk from algae because escape to environment. Several biosafety and environment risks are associated with GM algae. Related to the mass cultivation in open ponds, like horizontal gene transfer in cyanobacteria can cause gene transfer to their wild type relatives or other organism, as in literature has been reported. And in green algae, sexual reproduction in green algae with the wild type counterparts can easily transfer transgene to relatives, and there is a possibility of developing the new combinations. So these kind of hypothetical risk or the make the perception in public to go against these approaches. So here we have proposes that mitigation, how we can reduce the risk of the GM algae. Cyanobacteria is highly attractive organism for genetic modification because higher receptiveness of the foreign genes and associated, but this is highly associated with the risk of horizontal gene transfer. So how we can mitigate, we can grow it like an enclosed photobioreactor to reduce the mitigation, or we can reduce the dominance of GM algae by coupling with the gene of interest, which we do not want to make it dominant. Or we can link these GM algal with the specific nutrients, so only presence of these nutrients, these algae should grow. So these are the measurement we can take. And in GM algae, like green algae, prefer to reproduce asexually, so there is a gene containment. There is no fear of splash or contaminating other species. But in under drastic conditions, there may be possibility the haploid daughter cells may undergo sexual reproduction by forming gamete of plus and minus strains. So there are some mitigations we can take care of, like measurement, like again the closed photobioreactor or reducing the dominance by genetic engineering of this GM alga, or chloroplast transmission is the one possibility, because there is a metal inheritance of chloroplast gene occur in isomers green algae. And there's a, like under drastic conditions, the green algae splash is possible. So if we have a like buffer zone around our growing system ponds, then there will be least possibility of splash or spreading of this GM alga. So, many peoples may be interesting who controls these GM organisms or alga in India. GAC is the apex body under the Ministry of Environmental and Forest, who control these GM organism. If anybody interested, they have to go through with this regulatory agency. So these are the four different agency operating on different level. GAC is the apex committee, and DBT is the second most controlling body in GM, which is by Department of Biotechnology under Government of India. And the first level biosafety is like IBSC, which every institute implement these regulation. And ICR also has their own bodies which control the GM in their own agriculture systems. And there's a bill pending, the Biotechnology Regulatory Authority of India. This is the proposed bill is still pending from long time with the Standing Parliament Committee, which has not approved yet. Maybe one day it will approve, which will solve all the problems. So, so these are the more like policies, risk, and GM algae can be the knowledge can be get from this nice article written. Myth about GM algae. There's a myth that genetically transformed alga are more fit, more stronger than the wild type strain, and could take over the wild type population. As Dr. Steve Mayfield already shown that this is not the case. It has been GM alga spread up to 50 meter, but there was no harm to the wild type populations. So GM in
when you're turning, if you take the mic, otherwise it's not audible.
Okay.
When you're turning that side, use the mic.
Oh, sorry. So GM alga is less fit than the wild type due to transgene load, and it requires higher nutrition to produce more desirable products. So, so I don't think that GM alga is really going to harm the ecosystems. And moreover, GM alga or any alga grown in different system may not survive long in the new habitat. So I don't think that there's a concern. It's just perception of the public, just a myth. And GM could harm the ecosystem. No, because the genetic modification of algae is aimed for modifying either photosynthesis, biomass, or lipid biosin, not to generate harmful strain with respect to human health or disturbing the surrounding natural ecosystems. So this is I don't need to show this slide. Professor Mayfield already has covered his work. So this is about that there's no harm to the ecosystem. They have tested the EPA approved GM alga. And so the potential and limitation of using the genetically engineered microorganisms, there's a um, there's a report that algal biofuel production is neither environmentally nor commercially sustainable. Such kind of review article has been recently published in one of the three or four impact factor, has been highly highlighted by the media, because these are the attractive things for the media to make the perception about the field. Uh, but but if you look in nature, there is no robust strain of algae yet discovered from the nature that could be commercially viable. Cost is higher than fossils, so that's the one give this kind of concept to the different researchers. Uh, but overall, if we see that success of GM crop like cotton, soybean, corn, and higher yield of fermentation from genetically modified microbes may rationalize the use of GM approach to increase the product. So, not working, we not work only in GM field, we also go and lobby to make the people change the concept. This is Parliament member Dr. Sanjeev Balian from our constituency. I have talked to him, and then he talked to Mr. Prakash Javadekar when he was the environment minister. Then he made the written statement that there is no scientific evidence to prove that GM crop could harm soil, human health, environment. In the Rajya Sabha, he made the written statement, and last year our Prime Minister has tweeted that GM will bring the second green revolution after talking to Professor Deepak Pental. He's luckily my supervisor, or PhD supervisor also. And we also write commentaries about the GMOs. So I would like to thank Amit Kumar Singh, who has worked on this LCA and LCB work in the algae, and the Kunbi, she is a Nigerian girl who did some research work for 1 year in our lab, and Prachi also working on these heat-tolerant algae. Depo is again in Nigeria, came for small times, and Amita, she is currently in UK working on this genome editing in algal species, and Vipul Bhatnagar, he's working on different process studies in the algal using endophytic fungus, and these are like Parsoon, Devashri, Charlie, they all are involved in the different aspect of the of these genetic modifications, and we are thankful to our fundings from DVT, DST, NSF, Reliance, CPCL, Aban, Pan Ardes, and thankful to our current director, Dr. Dinkar Salunke, and Professor Maro Giga, and former director Professor Virender Singh Chauhan, and these are our current collaborators, UPenns, Ohio State, University of Arkansas, Reliance, CPCL, Aban, and University of East Anglia. So thank you. These are some foreign visitors who have visited our facility at ICGB, and there's some activities I have been involved organizing the retreat, yoga day, and thank you.
Thank you very much.